Viruses not only infect mammalian cells or human cells, but they can infect bacteria as well. Bacterial viruses are known as bacteriophages, and we saw these in the last section when we were talking about different structures for viruses. So they'll often have a very geometric head that will hold the genetic material, and then they'll have spike proteins where they can kind of grab on to the bacterial cell membrane cell wall and insert their nucleic acids, be it DNA or RNA. Most of them are going to be double-stranded DNA viruses, though there are some examples of single-stranded DNA viruses and RNA viruses as well. Now, you might think, hey, these are infecting bacteria, why do I care? But they do actually indirectly impact human health. If a bacteria gets infected with a viral nucleic acid, it can change their genetics, and it could help them or could hurt them. So it could make them less pathogenic, which is, hey, good for us. But a couple of them, it could make them pathogenic, and survival of the fittest, we could end up with more pathogenic bacteria growing and outcompeting the less pathogenic ones. Lysis is when you see the release of these viral particles. So if it inserts into the bacteria, it makes a bunch of copies, then often it will result in cell lysis of the bacteria, or it kind of splitting open and spitting out all of the viral particles. Bacteriophages often will take part in the Lytic lysogenic cycles. So these are two kind of connecting cycles that bacteriophages can use. Typically, we see the lytic cycle, and this is what we often think of with a viral infection, where you have your absorption and it will penetrate the host and it will copy its genetic material and assemble the new viron, and it will mature, release, and find new hosts, and it cycles around. But if the virus senses that the environment is not favorable, or stress or something like that triggers it, it can enter an alternative state known as the lysogenic state. In this case, the viral DNA can be incorporated into the DNA of the host, and it can kind of hide out there. And every time the bacteria copies its own genetic material, it'll copy the viral genetic material right along with it. It's not hurting the host. Other than that, it's just making it copy more DNA. And it can hide out here for years, potentially, until the environment's right. And then it can swap back into gear here and start copying and making more of itself. Okay, so lysogeny is known as the silent viral infection. Temperate phages are bacteriophages or bacterioviruses that can participate in either lytic or lysogenic cycle. So they can kind of swap between the two. These bacteriophages do not undergo replication or release immediately. So they could come in, insert their genetic material, hang out for a bit, and then start copying themselves. The DNA enters an inactive prophage state. So this would be when it's inserted into the bacterial genome and it's retained and copied with the host genome. Lysogeny is the condition in which the host chromosome is carrying the DNA. So if we go back a slide here, this would be lysogeny, where it has the bacterial genome in pink and it's carrying the blue viral genome. Induction is the process or the thing that triggers the lysogenic cell to become active and begin making viral particles again. Okay, so our next section, brief one covering how do we actually culture viruses, how do we study them in the lab? And one of the reasons that it's very difficult, and we're talking about this, is because viruses require hosts. So unlike bacteria, where I can take the actual E. coli itself and put it in some broth and grow it, viruses, we can't do that. If I put them in broth by themselves, they're not going to make it because there's no host. So they're a little bit trickier for cultivation. The primary purposes for wanting to culture viruses are to isolate and identify viruses in clinical specimens. If you want to determine what sort of infection is affecting your patient, you would need to isolate it and identify it. Another reason for viral cultivation is preparation of viruses for vaccines. So it can be whole viruses or attenuated viruses or viral components, but whatever it is, we have to prepare them and use them as vaccines. And then a third reason is maybe you just want to do research on them. You want to know, is it an enveloped or a naked virus? What are the sorts of genetic material that it has? Does it have spike proteins? What do they look like? What's the multiplication cycle? Any of its genetics? Well, to do that, you have to have the specimen to study. For a long time, live animals were used to study viruses. There were specifically bred strains of rats and mice, pigs, rabbits, all used to study animal viruses. Sometimes insects were used, non-human primates were used. It all depended on the host range because, again, if you're studying, I don't know, poliovirus, then you need to make sure that that virus matches the host that you're trying to study it in. And so host range has in a lot of ways held us back from big scientific discoveries because many viruses are very specific in the cell type or the species that they affect. Bird embryos or eggs have often been used as well. So you may have heard of this when you go in for a vaccine and they ask you if you have an egg allergy. Well, this is in large part because a lot of vaccines with viruses were prepped or grown in bird eggs. So a bird egg provides a nutrient-rich environment. It is sterile, and this all supports viral multiplication, replication. If you're trying to get lots of a specific viral particle, this is a great tool to use. In more recent years, we've moved to using more cell tissue culture techniques. So tissue culture is when you take animal cells and grow them in a sterile dish with special media to allow the cells to grow. So when I do cell culture, I might have, I don't know, mammary epithelial cells. And you'll have a little plastic petri dish that's been treated and sterilized for the cells. And you'll add the mammary epithelial cells into the dish, add media over the top of it. Often it's pink because it has a pH indicator to let you know if it's getting too acidic for the cells. It will have lots of nutrients. Often it's bovine serum in it, and it may have some antibiotics to keep it sterile and all of this. And you'll grow them in a dish at body temperature, often 37 degrees with a little bit of CO2 in the dark. And then when you look at them on the microscope, you'll see here, they're grown in a monolayer or a singular layer on the dish. And this is great for if you're trying to study viruses. It gets us a little bit further than whole animals in some cases. The downside with cell culture, though, is, as you're probably aware, we are not a monolayer of cells. I mean, all of our cells are organized into tissues and organs and 3D structures. And while this helps a little bit, it's not a perfect model for understanding the whole human body. Now, that said, it's a great tool for studying viruses. You can study viruses by adding them to tissue culture dishes to see how they affect the cells. Maybe you're looking for What sort of cytopathic effects? Is it a multinucleated cell? Does it have inclusion bodies? Well, you could add your virus to a cell that's in its host range and then see, oh, wait a second, it's lysing some of the cells and they're kind of losing their normal organized cell shape. Degeneration and lysis of host cells will indicate that there has been an infection. And since the cells are grown in a monolayer over here, you can look for plaques. Plaques are areas where there are clearings in the dish. So you would cover the entire dish with cells, add your virus, and then come back and look and go, oh, there's a hole there, there's a hole there, there's a hole there. There are 3 plaques or whatever it is to see how far the viral infection has progressed. This technique can also be used to study bacteriophages. So you can study and count the number of plaques on a mammalian cell culture dish, or you could grow bacteria on agar, add the virus, and you could see how many plaques there were for a bacteriophage. Okay, so our last section of the day is on non-cellular infectious agents. We have 2 main ones to discuss. The first one is a prion. Prions, again, are non-cellular. They're not living. They're just proteins or strings of amino acids that have gotten folded wrong. And as they're folded wrong, we know that structure determines function. And if the structure is not right, then they're not going to function right. And not only are they just non-functional, they can sometimes cause disease. So they can cause serious problems, especially with the nervous system. Usually it's progressive. So as you get one misfolded protein, you can get more and more and more, and ultimately you'll get to a tipping point where it is often fatal. Another non-cellular infectious agent is a viroid. These are much smaller than viruses, about 1/10 the size, and they're basically plant viruses, okay? So these act as parasites on plants. They do look a little bit different than what we traditionally think of as a virus. They don't have a capsid or a special coating. Basically, they're just naked RNA that can go and negatively impact the plant. Okay. A little bit more on prions. Prion diseases have been discovered in humans. One of the main ones is the Creutzfeldt-Jacobs disease. This affects the central nervous system. And as you get more and more of the misfolded proteins, they can gradually cause neural degeneration, and it can ultimately lead to death. Another big prion disease is BSE, or bovine spongiform encephalopathy, often known as mad cow disease. So this one is contracted by eating contaminated cow meat that has the misfolded proteins. You eat it, and then it causes neurological disorder issues, okay? So prions can either come from your own body, of you making misfolded proteins, or they can come from outside and be contracted as well. Right, so if you're interested in looking at some more important human viral families, we have this beautiful chart at the end of your chapter going through examples of DNA viruses and RNA viruses, many of which you probably heard of as like COVID-19, or maybe the human rhinovirus, dengue fever, Zika, all of these big human health issues. Not all, but many of them are connected to viruses. All right, so with that, that takes us to the end of chapter